to achieve after attending this lecture and reading your topic in the book? You should be able to discuss the determinants of bacterial pathogenesis, toxins, how they are produced, what are the types of toxins like exotoxin and endotoxin. You should also be able to discuss bacterial infections which are associated with cancers, describe the stages of infectious diseases, and describe the importance of Koch's postulates that we will do in the end. So to achieve these objectives, you have to be very alert, and I'm telling you in the beginning of the lecture कि आप लोग बहुत alert होके बैठे, ठीक है? इसमें कुछ ऐसी details आएंगी that you have to understand and make your concepts very clear, okay? So the toxins, we did in our previous lecture all the virulence factors that are produced by the bacteria that they use to produce disease. We saw that how they use the virulence factors, which are the capsule and the pili and fimbria and the various enzymes to invade the tissue. And through the invasion, then they cause inflammation that was of two types: pyogenic inflammation, that is pus forming lesions, and granulomatous inflammation in which granulomas were formed. So we did various virulence factors, but the main chunk was remaining that we are doing in a separate lecture, and that is toxigenicity. Yes, the microorganisms, they produce toxins. And toxins are very strong virulence factors that we will see how these toxins act on various tissues, cells and organs and produce disease. There are mainly two types of toxins, exotoxin and endotoxin. Exotoxins are secreted actively by the bacterial cell. Various bacteria, whether they are gram-negative or gram-positive, they actively secrete these exotoxins from their cells. While endotoxin is integral part of gram-negative cell wall, it is released only when the cell undergoes lessons, when it is destroyed. So please remember, each student of microbiology should remember all the time this basic fact that endotoxin is not secreted by the bacteria, it is the integral part of gram-negative cell wall. And it is released only when the cell is destroyed, when the cell wall is open, destroyed. Then exotoxins are polypeptides in nature, while endotoxins are lipopolysaccharides. So you see the chemical composition is also different. Exotoxins are heat labile, very sensitive to heat, while endotoxins are heat stable. They resist heat. Exotoxins are strongly antigenic and immunogenic. Now, in your physiology, you must have done immunity, basics of immunity. We will do it also according to pathological aspects. So, what is an antigen? Any foreign body that enters the body and an immune response is generated against it. That is an antigen. So, exotoxins are strongly antigenic. Immunogenic means that they give rise to a strong immune response. While endotoxins are antigenic, yes, but they are not very strongly immunogenic. So exotoxins can be used to produce toxoids and vaccines because they are highly antigenic, highly immunogenic, so we can use it to produce immunity through toxoid and vaccines. While endotoxins cannot be used as toxoids. Exotoxins have specific effects on certain cells and organs, while endotoxins exert generalized effects that we will see later. Exotoxins are produced by gram-positive and gram-negative bacteria both, while endotoxins are produced by gram-negative bacteria only. Why? Because they are the integral part of gram-negative cell wall only. They are not found in gram-positive bacteria. Exotoxins are encoded mostly by genes on plasmids and bacteriophages. So what does it mean? We have done bacteriophages multiple times before. We have talked about plasmids. So it shows that they can be transferred from one bacterium to another because they are present on, the genes are present on plasmids and bacteriophages. They keep transferring from one bacterium to another. So, and the endotoxins, they are encoded by genes on the bacterial chromosome. Now, as I told you that exotoxins are actively secreted. So there are various systems in bacteria for the secretion of exotoxins. There are basically 6 types of secretory systems that are known up till now. But the type 3 secretion system, which is also called an injectosome, is very important and very effective. So exotoxins can be secreted into the extracellular space, like outside the cells, and it can be secreted directly into the cell through an injection method, which we are calling the type 3 secretory system. And in this, an injection-like, needle-like projection, which is also known as molecular syringe, is there in the bacterium, and through which it injects its exotoxin directly into the cells of the host. So it is not exposed to antibodies in the extracellular space, and it is very effective. So this is how you can see, this is injection-like structure in the bacterium, and there is a platform-like structure. This is the basal body, this is the needle filament, and this is the portion from where this is the host cell. So you see, this is the bacterial cell. So how this is injecting its exotoxin in the host cell directly, like we inject something in some matter. Most of the exotoxins have 2 subunits, A and B. A is the active subunit. You can also remember by A for active and B for binding. A subunit is very active, and it is the main toxic material of exotoxin. And the B subunit is for binding. This is responsible for binding the toxin molecule to the specific receptors on the membrane of the human cell. And once it binds and stabilizes the toxin molecule, then it helps the active domain, the A portion or the A subunit, to enter the cell cytoplasm. So the subunit A, which is an enzyme actually, it catalyzes some reaction in the cell cytoplasm and exerts its toxic effects that disturb the function of the cell. For example, there is a reaction which is known as ADP phosphorylation, ADP ribosylation, sorry. ADP ribosylation means that there is an addition of the ribose sugar to any protein. And this target protein is affected by ADP ribose, and this process is known as ADP ribosylation. Now, if this protein is affected, then it will, its function will be disturbed. Either it will become inactive or it will become hyperactive depending on the situation. And this hyperactivity or inactivity causes the symptom of the disease. So is it clear up till now? The action of exotoxin, its 2 subunits, A and B, B for binding. A is the active portion, and it brings about changes in the cell of the host, that is the human being. We are talking about human microbiology or clinical microbiology. Now I will give examples, and these are given in your book, so you have to understand them, right? There are around 12 examples, so be very attentive, okay? Keep your mind very alert, okay? So number 1 is cholera toxin. I don't know how many of you have heard about cholera, but it is a very contagious disease. It affects the gastrointestinal symptoms. In this, the patient has, it undergoes profuse diarrhea and severe dehydration, and if it is not treated within due time, the person can die of dehydration and electrolyte imbalance. So the cholera toxin, this cholera is caused by a bacteria Vibrio cholerae, and this secretes an enzyme which is known as cholera toxin. It causes ADP ribosylation of a protein which is found in the intestinal cells, and this is called GS protein. This protein functions as a molecular switch, and it transmits signals from a variety of stimuli inside a cell to its interior, from outside to inside. So it gets activated, and then what happens that when the toxin causes the addition of ADP ribose to this GS protein, it suddenly becomes hyperactive, and it causes increase in adenylate cyclase activity, and in turn it increases cyclic AMP, and it results in production of watery diarrhea. Huge voluminous diarrhea is caused by this type of toxin. So this is how it works. This is, you can see that this is the B subunit, and this is going to get attached to this ganglioside receptor on the intestinal cell, and it is containing the A subunit inside. Once it gets attached, it will release the A subunit, and then it will go and bind with this protein, and this will cause increase in cyclic AMP and release of voluminous water with electrolytes in the intestinal lumen, and it will result in voluminous diarrhea. The diarrhea is very voluminous, and within a very short period of time, the person becomes dehydrated. And this infection is transmitted through contaminated water and seafood. This is Vibrio cholerae. So this is the bacterium that is the culprit that is secreting this type of toxin. Then another toxin is Shiga toxin, and this is produced by enterohemorrhagic strains of a bacterium which is called Escherichia coli. Now these particular examples you should remember. You will learn about these bacteria in detail when you will do systemic microbiology with systems, different systems. But here, because you have to understand the action of various exotoxins, and because you have to understand that there are so many varieties of exotoxin and each has a different function to do. And then we will compare with the generalized effects of endotoxins that are very few. So Shiga toxin is caused by, it is secreted by Escherichia coli with O157H7 serotype. This is a bacterium that causes bloody diarrhea, and this is associated with fast food. So be very careful when you go to fast food restaurants, especially hamburgers were implicated in this type of infection. In stools, blood is also fast because it causes hemorrhages in the intestinal cells. Again, it has two units. The subunit A, which is released inside the cell after the B subunit binds, it inhibits the protein synthesis, and it removes adenine from a specific site on the ribosome. So B subunit binds the toxin to the host cell, and when this protein synthesis is inhibited, the cells become necrosed. And not only the intestinal cells are involved, but it also has effect on the kidneys and the brain. So hemolytic uremic syndrome is also one of the signs of this type of diarrhea, in which there is renal involvement and blood in the urine also. So here you can see how this Shiga toxin is, first the one part is being attached to the receptor, and then it enters. The subunit is the unit which is binding, and then it goes inside a vesicle, and then the A subunit would be released, and it will act on a certain protein, and protein synthesis will be inhibited, leading to cell death. So the intestinal cell will start dying in necrosis, hemorrhages, and this function would be disturbed, and diarrhea would result. So these are some sources of this type of infection. Third example is botulinum toxin. It is a neurotoxin, and please remember that these toxins and this information is very important for you in your clinical um, trainings also, because you might come across cases that have infections, and some of these infections might be present there in the clinics. Botulinum toxin is a neurotoxin. It acts on the nerves. It's produced by the Clostridium botulinum. It is a bacteria which grows, which is anaerobic, and it grows in anaerobic conditions. So people who use canned food, and if the cans are not properly closed and they are broken, then this type of bacterium, they germinate and produce this toxin. So this blocks the release of acetylcholine at the nerve synapses. Now please remember your physiology and anatomy also. You have done neurology. So I will not explain in more detail, but you should understand that what happens at the synapses, and if acetylcholine is blocked, what happens? So it produces a flaccid type of paralysis. One polypeptide subunit binds by a receptor on the neuron, and the other degrades the proteins that are responsible for the release of acetylcholine. And it is one of the most lethal poisons. 1 microgram is lethal for humans. It's one of the most toxic compounds known. Please note it down and remember this. It's an important thing. So this is how the botulinum toxin acts. It inhibits acetylcholine. It's going here, this toxin, and it will block the proteins. It will disturb them that are going, their function is to release acetylcholine. So these proteins would be inhibited and acetylcholine would not be released, and the muscles will become inactive, resulting in flaccid paralysis. This is more detailed diagram for this. So this botulinum toxin is actually comes from eating improperly canned food. So that is how the gut is shown. They are absorbed through the gut, this toxin. But remember the rule of tissue tropism or cell tropism. It is coming from the gut, but it is attacking the nerve. So it will block those proteins that are responsible to release acetylcholine, and acetylcholine would not be released. And this is the improperly canned food items. You should be very careful when you use canned food. The fourth example is tetanus plasmine. Have heard about tetanus? Many of you must have been vaccinated for tetanus. It is included in the program of immunization. So it is a neurotoxin which is produced by Clostridium tetani, which is the causative agent of tetanus. And this neurotoxin is, the proteins are encoded by plasmid, and again 2 polypeptide subunits form this toxin. The heavy chain binds to gangliosides in the membrane of the neuron, and the light chain, which is a protease, it destroys the proteins. It degrades the protein responsible for the release of inhibitory neurotransmitter, resulting in a spastic paralysis. So both botulinum toxin and tetanus plasmine, both are acting on nerve endings, on the neurons, but their functions are different. One is causing a flaccid type of paralysis by inhibiting the release of acetylcholine, while the other is acting on the proteins that are responsible for release of inhibitory neurotransmitter, which is glycine. You can see here that this tetanus plasmine is going and acting on the synapses, and this inhibitory transmitter is coming, but these are blocking them so that inhibitory transmitter is not acting. So it causes spastic paralysis. The body goes into very severe spasms. Then the fifth example is the diphtheria toxin. Diphtheria is again infection most commonly in childhood, and the toxin gene is encoded by a prophage called corniphage. I hope that you remember bacteriophages. Okay, so the toxin enters the cell cytoplasm and inhibits protein synthesis, resulting in cell death. And there is one very prominent thing that happens in diphtheria, and that is formation of a pseudomembrane in the throat. So in my previous lecture, I told you when we were discussing pathogenesis that there are two types of inflammation in which pseudomembrane is formed. One was due to bacterium in the intestinal tract in colon, and it was causing pseudomembranous colitis. Remember? Prolonged use of antibiotics suppressing the normal flora, and um, this bacterium grows in the absence of these antibiotics and causes pseudomembranous colitis. In the same way, diphtheria toxin causes pseudomembrane formation. So I will tell you how the pseudomembrane is formed. Diphtheria is a contagious infection, usually of childhood, but since the vaccination has been started for this type of infection, its prevalence has decreased significantly. So Corynebacterium diphtheriae is the bacteria, and it produces diphtheria toxin. The B portion binds to the host cell receptor, and the A portion enters and it inactivates a protein which is called elongation factor 2. Now when this protein synthesis is prevented, the cell will die. And a lot of cells die and they become necrosed. And then what happens is that this type of pseudomembrane in the throat can be seen. If the doctor tries to remove this membrane, it bleeds profusely, which is very dangerous. Now, what is this membrane? All the collection of dead cell material, necrosed tissue, because the protein synthesis stops, the tissue from here is dying, the cells are dying, and there is a lot of cell debris, dead cells, and leukocytes, they all collect here, forming a yellowish gray membrane. And it makes it difficult for the person to breathe or even talk. Pertussis toxin, Bordetella pertussis is a bacterium that again it causes a childhood infection which is called whooping cough. Whooping cough is characterized by a cough which is associated with the barking or whooping sound. Why? Because the tissue in the larynx are inflamed severely. So when the child coughs and then tries to take in the breath, a very sharp uh, noise is made. So that is called whooping sound. Now this pertussis toxin, it again causes ADP ribosylation of a GI protein and it inactivates. The protein stops working. And inactivation of this protein turns on adenylate cyclase, and it is caused an increase in the amount of cyclic AMP, and it causes the symptoms of whooping cough. Severe inflammation occurs in the throat. So diphtheria and this pertussis toxin both act in the upper respiratory tract, and they inactivate the protein which is known as elongation factor, and it causes tissue necrosis. So you can see the airways are highly inflamed, and the sound which is produced is a sharp barking type of sound. Then there is one bacterium which is, I hope you are not bored, okay? But you have to tolerate this thing because this is very important, and you have to remember these 10 or 12 exotoxin actions. Bacillus anthracis. Anthrax, I don't know, you must be very young when anthrax was very popular in newspapers as a biowarfare weapon after 9/11. So anthrax causes an infection um, which causes lesions in the skin, and it can be inhaled also. So three types of exotoxins are produced by Bacillus anthracis. One is edema factor, one is lethal factor, and one is protective antigen. The edema factor, it raises cyclic AMP and results in loss of chloride ions in water and consequent edema also in the tissue. The lethal factor is a protease. When the word protease comes, remember that it will cleave a protein, or it will destroy a protein, or it will degrade a protein. So it causes failure of cell growth and cell death. And then there's a protective antigen which binds to cell surface receptors and forms pores in the human cell. And then edema factor and lethal factor enter the cell. They cause severe swelling, and this infection is acquired by people who deal with animal skin and animal hides, because this infection is common in animals. But if a person who is involved in dealing with the wool or the animal hides or animal skin, they sometimes get this infection. So the three factors that are the exotoxins, they cause severe swelling, edema, and consequent ulceration in the skin. Toxic shock syndrome toxin is produced by a gram-positive bacteria. And you see that the exotoxins are produced by gram-positive and gram-negative both. So it directly binds with proteins on the macrophages, and they cause overproduction of certain cytokines that are secretions from the macrophages. And these cytokines produce toxic shock. And it induces multiplication of T cells. So toxic shock is sometimes caused by contaminated padding, whether it is used to uh, heal some wounds, like a nasal bleeding sometimes happens, and pads are kept in the nose, nasal cavity. Or sometimes tampons that are used by females. They also, if they are contaminated, they can cause this toxic shock syndrome. Exfoliating toxin, it is also produced by Staphylococcus aureus, and it causes scalded skin syndrome, because this exotoxin, it cleaves desmoglin, which is a protein in the desmosomes of the skin. It acts on the skin and it results in detachment of the superficial layer of the skin. So the skin seems as if it is burned, you see. How many various types of exotoxin actions are there? Then there is panten-volatile leukocidin toxin. It is a pore-forming toxin. It is produced by a very resistant strain of Staphylococcus aureus, which is a gram-positive cocci, and it destroys the white blood cells, skin, and subcutaneous tissue, and causes huge, severe inflammation. So you see, it produces spores, this toxin, in the macrophages, and they burst open. It destroys the cells. Erythrogenic toxin is produced by Streptococcus pyogenes, and it causes a rash on the skin, which is called scarlet fever, fever with rash, and it acts as a super antigen. Now, exotoxins are also enterotoxins. We call enterotoxins when they act on the intestinal cells. These are basically exotoxins. So please remember, don't get confused by the name that enterotoxin is something else. Enterotoxin is not endotoxin. They are exotoxins in nature. But because they are acting on the intestinal cells, that is why we call them enterotoxin. Entero means intestinal. So they affect the enterocytes, and some food poisoning infections, they are caused by Staphylococcus aureus enterotoxin. Usually it happens due to barbecue food. Main symptoms are vomiting and watery diarrhea. It is also a super antigen. I will tell you what a super antigen is. Then there are enterotoxins that are heat labile, and they're produced by Escherichia coli. again A and B subunits, and A subunit mediates transfer of ADP ribose to a stimulatory coupling protein GS protein. It locks the GS protein in the on position and it stimulates adenylate cyclase to synthesize cyclic AMP. And you know what cyclic AMP does? Huge excretion of chloride ions and fluid and electrolyte loss. So the gut causes watery non-bloody diarrhea. There is a heat stable toxin A, and this is also produced by the same Escherichia coli that is not inactivated by boiling, and this stimulates guanylate cyclase and increases the concentration of cyclic GMP, and it causes diarrhea. So you see how many types of reactions these exotoxins cause. So now coming to the summary of what I have told you. The mechanism of action for exotoxins is ADP ribosylation. I told you what it is. Superantigen means they cause a very enhanced immune response. Then they cleave the proteins and they cleave other lecithin material in the skin. So these are the varieties of toxins produced by various bacteria. And you can go through this chart, and this is just summarized. It has summarized all the details that I have given you in this lecture. Now coming quickly to the endotoxin. We have seen that exotoxin has so many various types of actions. In some cases, it is increasing the activity, in some it is decreasing the activity of a protein. So there is a variety of actions. While endotoxins have very limited actions, they cause generalized effects like fever, leukopenia or leukocytosis, hypotension, decreased blood pressure, septic shock, disseminated intravascular coagulation. The blood starts clotting in the vessels, and necrosis and overall system failure. Now, macrophage is the major site of action. The endotoxin bind to the lipopolysaccharide binding proteins in the plasma. When the gram-negative bacterial cell bursts, the endotoxins are released and they bind with certain proteins that are LPS binding proteins. This complex then goes and binds on the surface of macrophages with a receptor which is called CD14. And the signal is activated and the macrophage starts to synthesize large amounts of cytokines. Cytokines are secretions from the white blood cells, especially macrophages. So interleukin-1 and tumor necrosis factor and nitric oxide, they are produced in a huge amount. This is how the endotoxin acts. It acts on the macrophages. Interleukin-1 is released, it causes fever, tumor necrosis factor causes fever and hypotension. They all act on the temperature regulatory system in the hypothalamus. Nitric oxide is also released, which causes vasodilation and hypotension. Then complement is activated. Complement system we will do with immunology. These are a set of proteins synthesized in the liver and they activate the tissue factor. Now, I have explained this in this chart. One thing which is important that I want to explain here is disseminated intravascular coagulation, because coagulation system becomes activated. So what happens that platelets start aggregating and the coagulation system becomes activated, and these coagulation occurs inside the blood vessels, which is known as intravascular disseminated intravascular coagulation or DIC. It's a hallmark of this gram-negative endotoxin action. So there is activation of alternate pathway of complement, activation of the macrophage, as I have told you. So then septic shock can occur. So bacteria are in the bloodstream, blood cultures are usually positive. It can cause death of a patient even if antibiotics have killed the bacteria because the toxin has been released. In toxic shock, it is the toxin which is circulating in the blood, and sometimes the blood cultures are negative. Systemic inflammatory response can occur due to sepsis, and sepsis is the result of these five major effects of endotoxin: fever, hypotension, and disseminated intravascular coagulation, effect on the macrophage, release of the interleukins, and the activation of the complement system. So it is also known as cytokine storm. This is how this gram-negative bacteria you can see. And now gram-positive bacteria also have element which is lipoteichoic acid. This teichoic acid also can exert the effects like endotoxin. So these endotoxins, they are going to attach to the CD40 receptor on the macrophage, and it will increase the secretion of human necrosis factor and interleukin 1, and it will act and on the blood vessels, will cause intravascular coagulation and direct tissue injury, which is the effect of the endotoxin. This is something for you to go through, and you will like to solve this type of quiz. So gram-negative, gram-positive bacteria also have teichoic acid that has endotoxin-like effect, but not all of them. Now, is bacterial infection associated with cancer? Yes. Two bacteria, Helicobacter pylori and Campylobacter, they belong to the same family, they are cousins, and they are associated with gastric carcinoma and carcinoma of the small intestine. And now coming to the last part, Koch's postulates. A scientist, Robert Koch, he designed these postulates to identify the causative agent of a particular disease. And he, according to these postulates, the microorganism or the pathogen must be present in all cases of the disease. The pathogen can be isolated from the disease host and grown in pure culture. And the same pathogen should be isolated from the pure culture that has caused the disease, and it can be re-isolated in the lab. So this is how it is called. The pathogen, like an animal sample was taken, and we see that this pathogen which is growing in the lab is from this horse, and these are the bacteria that are the same that entered the animal, that were injected in the animal, and they are isolated in the culture. Then if these bacteria injected to another animal, this culture from this dead animal from the lab will again give the same type of bacteria in the culture. It means that it is augmenting the presence of these bacteria. It is proving that the same bacteria were causing the disease in this horse that caused the disease in this lab animal. And these bacteria, when they are re-isolated from the new host, should be the same as in the original host. So this was like it was in the beginning of microbiology era, in which people were trying to solve the things that how this infection occurs, and can these pathogens be isolated? Can we know that what type of pathogen is causing disease? So these Koch's postulates are still they are popular, and they state that the same type of microorganism should be isolated from each case of the same disease. And when we take the sample and culture it on artificial culture medium, and then inject it into a lab animal, the same microorganism would be isolated. So still for the diagnosis of infection, we use the same protocol now, that we take the sample from the patient and we grow it in artificial culture in the lab, and then we identify the bacterium. Now in each case, we don't do animal inoculation, but yes, we confirm the growth, and we see that the same bacterium was isolated from the patient that was causing disease in other cases also. So with this, I finish my lecture, and I hope that you are not left mind boggled with this type of information, but please be very serious. Go through this topic in your book. It is a very extensive topic, right? And in a lecture, we highlight the important things that you should know. But you should be able to read in between the lines. What I have told you is to make it easy for you to understand, so that when you go through the lecture, you can understand the basic ones. The basic things that I wanted to tell you was about the exotoxins and endotoxins, the differences, the varieties of exotoxins. What is immunopathogenesis? This is just an effect of immune mechanism. The immune mechanism becomes active against some infection, sometimes it causes harm to the body itself. We know that immunity is for our protection, but sometimes when hyperimmunity occurs, when the immune response occurs, sometimes it causes harm to the body by forming cross-reacting antibodies. Means that the antibodies that are formed against the infecting agent sometimes start attacking the body's own cells. Then hypersensitivity responses are also harmful. Sometimes inhibition of phagocytosis occurs, and sometimes what happens that antigenic heterogeneity occurs, that the antigen sometimes resembles the cells of the body itself. So the immune mechanism starts attacking body's own cells. So we will do this